Welcome everyone to this week's edition of the Extracellular Vesicle Club. This event of the International Society for Extracellular Vesicles is normally hosted by myself, Ken Whitwer. I'm the president-elect of ICEV, and then I have a lot of good help from Clotilde Terry, who's the past president of ICEV, as well as Metka Lanasi, the chair of science and meetings for ICEV. And I would like to now introduce Carolina Balbi, who has kindly agreed to co-moderate this session today. Carolina is coming to us from the Student Network on EVs. So SNEV is an organization that was recently established just in the past couple of years. SNEV has its own leadership structure, has its own initiatives, and SNEV partners with ICEV, including on our annual meeting, and today on the EV Club. So every few months, we'll have somebody from SNEV coming in to moderate and suggesting an excellent paper like the one that we're going to discuss today. So Carolina, welcome. And I'd like to ask you to introduce yourself and then also to introduce our guest speaker today.
Sure. Thank you. Thank you very much. I would like to welcome everyone to this EV Club. As Ken said, this EV Club is now part of the International Society of Extracellular Vesicles, and today is in collaboration with SNEV. SNEV is the student network on extracellular vesicles. So if you are a student, an early career researcher, and you are working on EV and interested to join SNEV, so please don't be afraid to write to me or to reach us in the social network. We have Twitter, we have Instagram, and also Facebook. So there are a lot of events organized by SNEV that are really interesting. Today, I'm really happy to introduce you to Krishna, and Krishna is going to present his paper. He is now working in University of Ghent as a postdoctoral researcher, but Krishna has two PhD, not one really. So the first PhD was done in University of Azores, and the second at University of Ghent. In the first one, he got a PhD in animal reproduction, and the second in veterinary science. And now he's working on EV, and he's working on EV in big animal models, so not the classical model. And today he's going to present his paper on the regulation of the action from miR-378 that is enriching in blastocysts extracellular vesicles. So Krishna, thank you for today, and the stage is yours. You can share the screen. Just only one thing, if you want to write questions during the presentation, you can use the chat, and then I can read the question and ask you to unmute yourself.
Thank you, Carolina. Thank you so much. Can you see my screen now? Is it fine? Okay, I'll go ahead. Before going to my presentation, of course, yeah, I would like to thank Ken for these opportunities. Of course, also Tom yeah, they immediately responded when I sent this paper, and they accepted me for the presentation. I'm really honored, and I'm really happy that I'm presenting, because I was been waiting to do a presentation in Journal EV Club for a very long time, but finally this paper made it happen. So let's go to the presentation. Before I go with the paper details, so who are not familiar with IVF or reproduction or ARTs, so I'll take you to the journey of how assisted reproductive technologies work, and what are the pros and cons. So quickly I'll go through that. Then, so as for recent fact sheet of ESTRE, like European Society of Human Reproduction, so 1 in 6 couples are facing 1 in 6 couples are facing reproductive infertility in any time of their reproductive lifetime. So you see that these problems are caused due to different it could be it's a multifactorial, I could say, like it could be related to the lifestyle and habits and a lot of other things involved in the infertility. When the ARTs step in, that is assisted reproductive technologies, that was first IVF baby was born like in 1978. Ever since ARTs came into play, there was a lot of babies were born with the help of with the ARTs. So around like So far, around 9 million babies are born with the ARTs. Of course, most of the ART treatments are given between the age groups of 30 to 39 women. So, of course, there are a lot of there are a lot of pros and cons with the ARTs. So I'm going to explain what are those. So as I explained in the human, ARTs are also active after the discovery of IVF in humans, and there is also in bovine, as I'm currently previously working on bovine model, and of course this paper is also on bovine model. So in bovine, the first calf was born in 1981, and then followed by birth in pigs and also in the lambs. So there was also around like 1 million of cows were born with the help of ARTs with assisted reproductive technologies. Indeed, as I explained, that there are several challenges that are in the assisted reproductive technologies. Despite the number of IVF and like in vitro fertilization, intracytoplasmic injections that take place, but still the percentage of take-home baby, that means the pregnancy and delivery of the baby, are quite low. As you can see, it's around like 30% in the older age women, like when they're on about 35 years. When they're below 35 years, they're around like between 30 to 49%. So still the take-home baby rate was quite low. That means the pregnancy, not only the like if you exclude the miscarriages and other things, and the take-home baby is really counts, you know. So there are several challenges. One of the thing of the most people struggle still is like choosing a right embryo. There is importance of the viable embryo. So when the embryos are developed in vitro conditions, so it's important to see their based on the morphology and the embryo kinetics are the very important things that choose a perfect embryo that can be transplanted, and then they can use an implantation. But indeed, there are a lot of contradictions, like whether the good quality embryos or poor quality embryos give the implantation or not, or give the pregnancy or not. It's still not concrete because it varies, because even the poor quality embryo could give a pregnancy, or a bad quality cannot, good quality cannot deliver a pregnancy, because it's all depends on multifactorial things. It's mostly connected to the implantation of the embryo to the endometrium or uterus. So so as I say, like we cannot judge based on the morphokinetics or the embryo quality. It's all depending on how the functionality and the communication between the crosstalk between mother and the embryo. So I would like to explain what is implantation, or the process of implantation. What is a window of implantation? As you can see that when the implantation process takes place on the day 5, and they usually in the human culture system, they use embryo that is transferred usually on day 5 or day 3 of the embryo culture media. They transfer it to the patient, and the implantation takes place on the day 5. Even in the in vivo process, it also happens on the day 5 of the embryo development. That is in the when the embryo is in the reach of in the near to the hatching rate, or in the blastocyst stage actually. So usually the interaction between endometrium and the embryo is very crucial, because the endometrium, when the embryo enters the uterus, and then it starts preparing itself with a lot of hormonal changes, and it will help, and it creates a lot of progesterone, and also embryo needs to hatch. So as you can see from this on the image, the embryo that is going from the stages of from the blastocyst stage to the hatching stage. So that is a hatching stage that you see that the zona of the embryo is out. And then the embryo, or the hatched embryo, is ready to implant. The connection between the endometrium and the embryo is the hatching. When embryo hatches, so the trophectodrome is exposed on the surface, is called trophectodrome. So it will attach to the endometrium and the cell adhesion molecules, which plays a crucial role. So when that helps the embryo to attach to the endometrium, and then it is called the process of implantation, where the embryo or a hatched embryo attached to the endometrium. So the hatching is a very crucial process. As I say, that it helps to the binding of endometrium. So as I said, the implantation, embryo implantation success or failure could be multifactorial. It could be maternal aging, lifestyle, and infection of the of uterus, like endometritis stuff. And of course, the success is mostly, it is also depending on the endometrial reactivity, the transfer of the embryo, when the embryo is transferred, whether how the embryo is transferred, and most crucial is the embryo hatching. So because despite the fact that they're the in vivo developed or in vivo developed embryos has the capacity to implant easily, but in case of the in vitro developed embryos, the implantation success is only 30%. So as I said, that there is only 30% chances before too. So there's a lot of responses with for the embryo to implant. Just one of the major factors is the embryo hatching. The hatching of embryo is important to attach to the endometrium. So of course, when there are other factors also that comes to the failure of the embryo implantations, like for example, like immunological factors and other hormonal factors also impact on the hatching or not a hatching, on the implantation. So as I said, that embryo hatching is important. So let's go into my presentation. I'm going to talk on my recent paper that was published in PNAS, which is where hatching is regulated by the microRNA 378, and which is derived from extracellular vesicles, which is also extracted from the blastocyst conditioned media. So before going to this topic and how our research lab ended up into extracellular vesicle research, so I need to explain a little bit briefing on that. Back in 2011, we use a routine in vitro culture system. Usually, most of the things that I'm explaining here are in a bovine system. I will explain in the next couple of slides why we choose bovine system. So in routine culture systems, we use we collect the ovaries from the slaughterhouse of bovine ovaries, and then we aspirate them and we mature them in in vitro maturation media, and we also take the sperm from the for the containers where the sperms are stored in the cryopreservation, and we fertilize them, and then we put all the embryos in the group culture system. So this is very important that we do. Why we choose to use the group culture system in bovine, and why not the individual culture system, which I will explain in the further slide. So we initially back in 2011, we are focusing on only the group culture system we have, because the major reason is that we have higher blastocyst rate compared to the individual culture system. But the reason is quite unknown at that time point. But we changed, replacing, like, for example, as you can see from this slide, in this paper, which was published back in 2014, where we tested replacing the media, like replacing the serum with BSA and ITAs, and we could see that there is a higher blastocyst development rate in the group culture system and also in the individual culture system. However, we are not sure what are the crucial molecules that are helping in the group culture system, not in the individual culture system, because still the individual culture system in bovine was still 30%. So they're not optimal of 50% or above 50%. So we, and back in 2015, we wrote a review paper where we discovered different autocrine factors released by the embryo when they are in the group culture system, and when they're cultured in the single or individual culture system. One of the crucial factors that we identified was extracellular vesicles. So that is our triggering point that we came into extracellular vesicle research. That is the point when I joined the Anwan Sum lab, where I started working on extracellular vesicles. So yeah, as you know, this is the statistics that I took from Roy et al., which is published in a International Journal of Extracellular Vesicles, where they saw there is a significant impact in growing of extracellular vesicle research in terms of exosomes, microvesicles, and other things. And however, most of you are familiar with what is the biogenesis of extracellular vesicles, but I'll briefly go through that. So we usually extracellular vesicles are broadly classified into 3 types: exosomes, microvesicles, and apoptotic bodies. And however, still the origin of the extracellular vesicles is not quite yet known. They come from the late endoplasmic reticulum and form into the multivesicular bodies, and they're released through the plasma membrane. And of course, there are different sizes of the extracellular vesicles. Depending on the size, they're coined as exosomes or microvesicles. However, the biological function of the extracellular vesicles are broadly broadened in the different areas. It depends on the origin of the extraction procedure or different biological fluids. The content of the EVs varies. That is what we also identified in our work. So the nature of the EVs that mimic the cellular components in the smaller miniature level, like microRNAs, DNAs, and nucleic acids inside of it, and carry this crucial information to others. So we came up with the first research questions that we have when we started working on extracellular vesicles. Like, as I explained, like, we focused initially on the group culture system because group culture system has a higher blastocyst rate compared to the individual culture system. So we thought, in the group culture system, maybe embryos are released between them and they have a higher and better communication, and they're improving the embryo quality there. And also we saw in the embryo quality compared to the individual system. So first hypothesis, we thought, like, whether embryos release the EVs or whether the embryos can uptake the EVs or not. Of course, embryo quality of the EVs can affect the characteristics of the EVs. This is what our major focus happened later on, which is also showed in this paper too. So for basic of these 2 research questions, we published 2 articles back in 2018 showing the extracellular vesicles are released by the embryo, and they're also taken up by the embryo. You can see that this is a blastocyst stage embryo where the EVs are labeled with PKH labeling. They are uptaken by the embryo, and they are inside the embryo. And this is a stack image of the embryo. So you can, it's a 3D structure. You can see that one important thing that we saw, that these EVs are mostly in the inner cell mass, where the baby, where inner cell mass in the future turns into the baby. That's what we mainly saw, that most of the EVs are inside on the inner cell mass range compared to the other part of the embryo. So this is the image, and we also find the key findings in this paper also that we showed, which I showed which I did not present in the presentation, of course, because we also saw that the BSA, that molecule that we are supplementing in the in vitro culture system, and that fetal bovine serum already have EV, so we replace the fetal bovine serum with bovine serum albumin with particular of this code from Sigma. So we identified whether this BSA molecule have EVs or not. So we saw that they happen to have some other things. They are probably albumin molecules, and we tested with the immunogold labeling with this BSA added to the culture media, and we did not find any EVs there. They're rather other lipoprotein molecules released by them. So one thing we showed that this BSA that we are supplementing to on behalf of serum or fetal bovine serum, that it's not having any EVs there. One more findings that we saw in this paper also, that EVs that are collected from the group culture system supplemented to the individual culture embryos had a higher blastocyst development rate. So we concluded this paper that showing that EVs has a functionality impact and also has a developmental rate, which is improving the quality of the embryos. So that means one of the crucial molecules that we thought earlier was the EVs that are carrying messages between the embryos. So why we choose a bovine model, as I say that because it's easy to mimic the human cultures, human embryo culture system. So we in bovine we just cultured them as in humans. We picked the bovine model because as I said, that it's easy, and then we just culture them in individual droplets, in the culture droplets. So here comes the major two questions that, as I said, that whether the embryo quality, like for example, if it's a blastocyst, the EV's quality, EV's sizes differs, or EV's content could be differing, or whether firstly whether we can isolate the EVs from that or not. Because when we started with the group culture system, we struggled a lot in optimization because it requires around a pooling of large amounts of conditioned media. We need to culture around 1,000 embryos to get the pooling of the media, and then we could isolate the EVs. Similarly, for the individual culture system, it would be much more difficult and much more challenging for us. So, but we overcome this challenge by using different techniques, of course, which I'm going in the further slides. So to work out this hypothesis, what we thought, like, so as we said that we use routine in vitro culture systems, like as I showed in the earlier slide. So we take the ovaries from the slaughterhouse and we fertilize them. We instead of doing the group culture system, we are doing the individual culture system. In bovine, the major difference in the human and bovine is, like, in human they culture the embryos until day 5. In bovine, you can go until day 7, day 8, and also until day 11. It can reach the hatching stage. That's the advantage, that you can culture more and then you can get more information. So, but in our lab, we only do until day 8. So this individual culture system droplets, as you can see in the experimental design. So we thought of isolating the conditioned media first based on the developmental stage. So on the day 8 of the culture system, we check the embryo quality, whether it's a blastocyst, whether it's not blastocyst. That means it could be arrested at early stages of the embryo development, like 2-cell stage, 4-cell stage, or it could be arrested at the morula stage, that is like 16-cell or 18-cell stage. So we pool this media and we try to isolate the media. We try to isolate the EVs from that and find the potential microRNA there. So that was the hypothesis of this. So to work out this plan, firstly, we need to isolate the EVs. So firstly, we try to isolate the EVs with eyes on columns with the size exclusion chromatography method. During our optimization of the several optimization techniques that we used initially that I showed in my papers and first 2 papers, we figured out that if I want to isolate EVs from the conditioned media from the lower concentrated samples, size exclusion chromatography was a little bit efficient in isolating. Of course, there is no perfect technique, and so far, no concrete technique that in isolating the EVs. So we use these eyes on columns that's supplied by eyes on company. So we use a single column method, single size exclusion chromatography method, where we take a group of divided into blastocysts and non-blastocysts based on the development quality. And then we followed the supplementation and adding the conditioned media on the top of the ISON, and then we use these Amicon ultra filters to concentrate the samples, and finally we have 200 microliters of EV pellet from this initial run of isolating and characterization of EVs. So for initial pool, we need to culture around 120 presumed zygotes, which is 3 biological replicates. We need to pull them, and we separated them with blastocysts and non-blastocysts, and we could characterize them with 3 different techniques there. The standard MISF guideline techniques. These are the results we found with the Western blotting, electron nanoparticle tracking analysis, and also electron microscopy. And of course, we did not only the EV specific antibodies like CD63, CD9, TSC101, but we also tested the fractions of the ISON columns with what are the fractions that we isolated. Like 6 to 15 fractions is the ones that we isolated the EVs, and then we pooled these columns for concentration step, and we find that other non-EV specific antibodies like AGO2 and apolipoproteins are expressed mostly in the later fractions of the EV isolation. So we saw the presence of these antibodies, and we also see there is bigger sizes of the EVs in the blastocyst derived conditioned media, whereas in the non-blastocysts they are relatively smaller compared to the blastocysts, but this is not quite concrete enough to show that there is a difference. But of course, you can see the sizes are differing when this blastocyst conditioned media derived EVs and compared to the non-blastocysts. Of course, the particle concentration was higher in the blastocysts because I see that not relatively that significant enough, but they are relatively higher in the blastocyst EVs because they are probably in that stage of higher development stage, they are releasing a lot of EVs there to communicate with the endometrium. But this is one way of identification or differentiating the EVs derived from blastocysts and non-blastocysts. But for the next level of EV isolation, we need to go through a lot of struggle in optimizing or isolating the EVs from lower volumes of conditioned media, or I would rather say less concentrated media compared to high concentrated conditioned media. So we need to concentrate our samples. So we need to pool a lot of media to get a reasonable amount of total RNA, which we can do the sequencing on that. So for this purpose, we need, for example, we need to culture around like 2,000 presumed zygotes. That is quite a lot. So we did like 9 biological replicates here. So we divided them based on blastocyst and non-blastocyst group. For example, for one replicate of we take here in this picture, we take one replicate, that is, we use 2 ml of conditioned media, and then we split them into 4 different groups, as you can see in the picture. And the 4 different groups are divided into the 500 microliters, because with this columns that we can use, the maximum volume is 500. So we use the 500 microliters of conditioned media for one isolation. So we did for every 500. So in the end, we pulled a lot of EVs there, and then we did the concentration step. In the end, we could achieve the concentration step of EV pellet of 600 microliters. And then we could do the total RNA extraction with Norgan Biotech kit, which is also in collaboration with ISON columns. And then we could able to do the bioanalyzer first, and then we further went with the sequencing. During the bioanalyzer results, as you can see, we have 6 biological replicates from 3 biological replicates from each group. So the concentrations are around like in ranging of 100 to 300 picograms per microliter, which is the bare minimum volume that required for microRNA sequencing is 100. So we are in the bare minimum range, but they are not concrete enough. But we saw one sample, which is like, for example, non-blastocyst derived sample D, which has higher peaks, and then which we came to know there's a lot of degradation happened in the sample. However, other samples are relatively fine. So we continued our sequencing experiment, and then we were able to differentiate these groups of non-blastocysts and blastocysts EVs derived microRNAs. And we could identify in total 69 differentially expressed microRNAs. In these 69 differentially expressed microRNAs, we identified 48 novel microRNAs, which are not yet known, and then 21 conserved microRNAs, which are already known. And then we choose one specific microRNA. Before choosing one specific microRNA, why we choose, and I'm going to explain this next slides. So we did a gene target analysis. I had a great help, my colleague Tim, who did a lot of statistical analysis for me, and he's also one of the co-author of this paper. And he came up with a simple enrichment analysis from this gene target analysis, which is a better way of presentation. I see that it shows based on GoKEX pathways enrichment analysis. In this simple enrichment analysis, you could see that there are there are a lot of targets, and then you can see the rightful amount of signaling pathways that are connected to these differentially expressed microRNAs. They are all, as you can see from all the three graphs, and you can see they are mostly connected to the membrane binding regulation pathways, cellular development. They're mostly connected with this. So we, from these connected pathways, we also did one gene target analysis on this specific microRNA, because we saw this specific microRNA is also highly expressed in the blastocyst EVs. We saw that maybe we can test this functionality of this microRNA for further analysis. And we saw that they're mostly connected to the cell growth, cell proliferation. With the gene target analysis, we could identify that. And I would like to quote some of the papers that we found interesting back in before going to our studies. We saw these two interesting papers, like this miR-378, 378, which is also a precursor in the human samples, which has a similar nucleotide base pair range with a human precursor. So we choose this microRNA because this has a really an impact on the cellular regulation, cellular migration, also connecting to the embryo quality and development. So we choose this microRNA for functionality testing, but before going to that, we did an additional experiment, because during our process of RNA isolation from the EVs, we did not treat the EVs with any of the protease and RNase treatment. So we want to make sure. So we did an additional step of EV isolation from the blastocyst condition media. So again, we used a lot of embryos to culture them and pool them, and we isolated the EVs from the same technique. And then we treated the EVs with protease and RNase, and protease, RNase, and detergent, to see whether the EVs, microRNAs that we identified, whether they're coming from surface or not. But we specifically tested only these specific microRNA. So, but we observed one specific thing in this process. So we used two different samples here. So one samples are EV, blastocyst derived EV samples, which are undergo this treatment of control protease RNase treatment, protease RNase, and detergent. But we also tested directly the RNA from the condition media, from blastocyst condition media. We saw a different thing, that here, when we isolate the total RNA with these treatments, and there is a drastic decline in the total RNA concentration. So it's immediately they're very lower when we use RNase and protease. That means they are not protected. Of course, with EV samples, we don't find any difference until the detergent is used. However, yeah, we also see that microRNA 378 is expressed even though after the treatment of RNase and protease. So this is specifically that microRNA 378 is originated from the EVs. And further on, to test the functionality of the microRNAs, we want to test whether the microRNAs that we get from commercially from the companies that designed microRNAs, whether they can penetrate to the embryos or not. Because like a lot of researchers are using transfection reagents and also intracytoplasmic injections to transfer these microRNAs. But we want to make sure, we want to make the process easy, whether we can try directly the microRNAs, because they are very small in size. And of course, it's important to know whether they can penetrate through the zona of the embryo, because zona is a protection barrier of the embryo. It's difficult. Even viruses cannot penetrate the embryos. So we want to make sure whether the microRNAs that we add to the culture media can penetrate or not. So we used labeled microRNAs of 5' FAM labeling, and then we cultured them on the day 2 of the on the early stage of the embryo development, like on day 0. And then we cultured until day 8 with this labeled microRNAs. And we could clearly see that labeled microRNAs that can penetrate through the embryo with the zona. And you can see in this video of the microRNA mimics, and in this embryo only, you can see the blue fluorescence is the Hertz staining, where you can see the nucleus. And whereas in the green fluorescence and blue fluorescence, you can see on the microRNA mimic, where they can go inside the microRNA, where they can go inside the embryo, even with the zona intact. And we also see, this is another image where showing 3 different segments of and showing that microRNA mimics labeled ones, they can penetrate through the embryo. And we saw this stability of this microRNA from day 2. Day 2 is a cleavage stage, as you can see on the cleavage stage, they can penetrate until the day 8, where in the bovine we culture until day 8. We could see that microRNA mimics are inside the nucleus of the embryo, and they can penetrate into the embryo. So this is one of the crucial finding of the paper, that we saw that we don't need any necessary reagents of transfection reagents for the penetration. So rather they can go, but we tested in the recent findings in our lab that BSA that we add supplement in the condition media that helps in the transport of the microRNAs into the cells, because BSA is also a cargo carrier molecule. It's also known that it binds to the lot of lipoproteins and stuff. So we thought it also binds to the microRNAs that we also saw in several review papers and also in the research papers, that the microRNAs that we supplement can also helps in the transportation with the help of BSA. That's what we saw in our recent research finding, but we're still ongoing research is going on in that area. So further on, so as I said, the microRNA mimics that we supplemented, they can penetrate. So we thought we added this designed miR-378, 378, which is we added the mimics and inhibitors to the condition media on day 2 of the group culture system. Why we didn't test? We did not test in the individual culture system, because the concentration will be too low for the individual culture system, because it's a single droplets and it's 20 microliter droplets, whereas in the group culture system is 50 microliter droplets and you have a lot of embryos in it. So there is a high chance of penetration there. So we used mimics and inhibitors, and we checked the embryo quality and embryo development rate, and we also take the embryos on the day 8, which are blastocysts, for transcriptomic analysis, how they are influencing on that. So we saw a phenomenal change when we add these microRNA mimics of 378. As you can see, there is a higher hatching rate. As you can see, there is a higher hatching rate here of 46%, which is phenomenal compared to the control sample. And we saw when we add this mimic on day 7 itself, we saw the lot of embryos reaching to the blastocyst stage. So that was very high range. So that is a crucial thing that we saw compared to the control sample. There is a higher hatching rate. The hatching rate is here shown is based on the day 8 of the embryo development from the culture. So we calculated this based on the day 8. And we also saw that there is a higher total cell number. So we did a embryo staining, differential staining analysis, where we count how many cells are there in the embryo and how many inner cell mass and protectome are there. So we count them, and there was also a higher range. There is a significant difference between the control sample compared to when we supplement this with miR-378, 378, and you can see that there are higher percentage of total cell number and also inner cell mass ratio, which is very important also during like we need a viable embryo for the implantation. So, and on contrary, when we add the inhibitors of this miR-378, it's perfectly, you can see it's a contradictory result. We saw a lower hatching rate and also lower development rate. It was clear, and there was significant difference actually. And apparently, it's also reflected in the embryo quality. We saw that the total cell number was quite low compared to the control, and also protectome cells are low, and also inner cell mass ratio was low, and also apoptotic ratio was really high. You can see a huge difference between the control. So we saw that when we add this miR-378, 378, it tend to regulate the hatching process and also the blastocyst development rate. So far, we saw this on the embryo development and embryo quality level. Of course, we also did transcriptomics on this mimic derived blastocysts and also inhibitor derived blastocysts. And we could see that in total, there are around 68 differentially expressed genes. So in these, they are perfectly upregulated and downregulated. In these specific genes, we identified several of the genes are related to embryo quality and embryo development. You can see that based on the simple enrichment analysis, they're again controlling the regulation pathways and the metabolic pathways connected to the embryo quality and embryo development. And of course, you also see that they're mostly connected to EVs are derived, as they also see in the endosomes and extracellular matrix complexes. So in the end, we also see specific genes that are regulated by this microRNA, addition of this microRNA. So in conclusion, what we say that for a hatching to improve that microRNA 378, which also has a precursor in human samples, can be one of the solution to help the hatching. I forgot one point there in the introduction. As I say that hatching plays a crucial role. Of course, there are different techniques to improve the hatching. There are assistant hatching technique and laser dissection technique where to improve in the human samples to attachment of the embryo to the endometrium. So here we see that microRNA 378, as in bovine, which is having a perfect precursor in human samples, has a close connection in controlling the hatching rate, which can improve, which is also crucial for embryo implantation. So that's how we ended up with microRNA 378 can regulate the hatching. However, we focused also in the human data on this specific microRNA 378 we're currently going through. So we collect the spunt media from pregnancy and non-pregnancy women, and we collect them. We initially did a trial analysis, and we currently it's a solid research that I'm doing on the pregnancy and non-pregnancy women. So we collect on day 5 of the condition media. We collect the pregnancy and non-pregnancy condition media, and we check. We did same procedure of isolating the EVs and total RNA, and we saw that implanted EVs and non-implanted EVs are on the similar range of the sizes, and you can see in the electron microscopy imaging the sizes does not differ much. And this is an immunostaining image of CD9. This is not gold labeling technique. It's just immunostaining where we use CD9 antibody, and we could see the presence of the EVs, which are completely surrounded by antibodies. You can see the black stainings of the CD9. Of course, this is one way to prove also that EV presence that can be isolated from the implanted and non-implanted samples, or you can say pregnancy or non-pregnancy. Currently, we have some interesting results that I'm ongoing. So we are trying to figure out a potential microRNAs that can be derived from the condition media that can be used as a biomarker. So we can see that potential of the EV microRNAs can regulate not only the embryo development, it also can influence it can show the path of biomarker availability. So we're currently doing this research, and we have some interesting findings. But soon, maybe the next journal club, when I publish this paper, I'll talk more on that. But the take-home message is that so EVs that are derived from the condition media of the spunt media, from either from the humans or the bovine system, we can identify, we can make biomarkers out of it. Like it's all, we can use it as for functionality testing. That is the beauty of the extracellular vesicle research, that I saw that the cargo content that carried by the EVs are very crucial, because that is really triggering the mechanism. And also focusing on the endometrial cell culture could also give a new findings on the finding of biomarker. So yeah, everyone is focusing on this research. Like ever since the discovery of IVF techniques, there have been a lot of researchers focusing on finding the biomarkers using taking the condition media and taking other things to find a specific marker to identify. But yeah, still it's ongoing. But EVs could be a solution in finding a biomarker. So I would like to conclude my presentation with acknowledgements. I would like to thank my team and particularly my colleague from pharmacy department team who helped me with the bioinformatic analysis, and also Anne Hendricks, which we are been collaborating ever since I started my PhD, and currently also as a postdoc. Yeah, she was great, helpful with a lot of techniques, information. Yeah, that's all for me. Please, if you have any questions, just post me.
Thank you, Krishna, for the presentation. It was really nice. Has someone have some question, or I can start with one? I was curious if you have it, because you tried to. Okay. There is someone with the microphones which are done. No. Okay. You treated your embryos with the microRNA, but did you try also to treat them with the extracellular vesicles? So if extracellular vesicles from blastocysts are enriched with the microRNA, did you try to do this experiment?
Yeah, excellent question. Yeah, we initially did that experiment actually. Unfortunately, we don't see because the concentration is not quite enough as microRNAs, because with microRNAs you get higher concentrations you can add, but with EVs you need to pool, as I did with the RNA sequencing. So you need a lot of embryos cultured, and then you pool that. That could be tried, but we did not do that much large scale experiment again. Maybe that could be a possibility to improve also. But we saw rather in my first paper where I could show that EVs derived from the group culture showed an influence on the individual culture. We saw an improvement there. So definitely EVs have functionality improvement there, not just a microRNAs.
Yeah, sure. Yeah, sure. It was the starting point of your observation. Yeah. So, of course. And I have another fast question. You show that there is a difference in size also in the vesicles between the two groups. Do you have any explanation of that? Because it should be.
Yeah, I forgot to explain that. Yeah, thanks for pointing out. Yeah, because when the embryos reach to the blastocyst stage, so the pore size of the zona was much bigger than when they're in the initial stages, like in the 2-cell stage or 4-cell stage. So the release of the EVs, or if there is microvesicles, they're budding out. If they're from exosomes, they're released through the plasma membrane. So probably that's our hypothesis, that they release the smaller EVs when they're in the early stage of the embryo development, but when they reach the higher hatching stage or higher embryo development stage, they could release a bigger size particles. That we specifically saw that actually also in the human samples.
You didn't find it. I know it wasn't the last point of your work, but you check if there are some markers, different markers. So if they are reached in microvesicles in one part and reached in other markers in the other population of vesicles.
Yeah, we didn't focus on that. Yeah, of course, it requires we hardly struggled with this experiments too, because it requires a lot of pooling there, and a lot of less researchers are working on focusing on that, isolating the EVs from these lower amounts of conditioned media, and it's quite maybe. Yeah, I think that's a good hypothesis that you say. Maybe that could be gives more research opportunities, like dividing the EV population, the small EVs and large EVs could show some difference actually. And the microRNA content, yes.
Yeah. Okay, so Ken, do you have some questions?
Yes, I do. I have a few. So Krishna, thanks so much for the presentation. This is really educational for me. And it also raised some questions that I realized I knew nothing about. So very practically, when there are multiple embryos that are being cultured, are they cultured together? That is to say, with the same
group culture. Yeah, that's what we did. We did all the individual culture systems. So every embryo is cultured single droplet. Pooling them in the group culture, it's not a good suggestion, and you need to separate them at one point, and still you have a crosstalk between. So I don't encourage in the group culture system. But
is that how it's done normally in human?
No, they do in individual system, mostly individual system.
Okay, got it.
In bovine, they do a lot of group culture, but
Yeah,
some labs they prefer to do in humans also.
yeah.
In some labs prefer to do group culture because they're higher blastocyst development rate.
Okay, got it. Okay, so normally it is done individually.
Individually, yes.
In the clinic. Okay, got it. Very good. So the other question, actually, I have a couple more questions, but one of them is about the binding partners of the microRNA. So and I'm sorry if I missed this, but perhaps you could enlighten us on whether you have a precursor microRNA that's there, or whether you have the mature microRNA that's found in the EVs. That's the first question, and then secondly, is that microRNA, if it is the mature microRNA, is it bound to an Argonaut? And have you looked at Argonaut levels?
That's a very interesting question also for me too. We did not focus on we saw the premature microRNA, also mature microRNAs in our samples, but I did not see on the binding level. Indeed, looking into the binding level also give a new perspective. But we saw in particular in the sequencing analysis, we only saw the microRNA that we found in because we extract the total RNA. We also see in the different RNAs, but there are more circular RNAs and other transfer RNAs. But there is higher content of circular and transfer RNAs in these samples actually. So but with the binding thing, we saw recently. We saw some interesting findings when we want to try this same thing in the oocyte level. They cannot penetrate to the oocyte. There is no penetration of microRNAs there. But when we add BSA or any other molecule, like mostly the binding molecules like BSA, it helps in transfer of these microRNAs into the embryo, not the embryo, in the oocyte level. But embryo, it was quite easy because we are already using BSA in the conditioned media. So that's what we saw a difference. When we leave out the BSA or the serum, there is no transport mechanism of the microRNAs there. That maybe we are still need to work in progress on that because BSA could be a binding molecule to help the transfection. We don't need any transfection reagents there. That's what I think.
Quite interesting. Yeah. So how do you think that is being transferred? Then how do you have an intriguing hypothesis for what's behind that?
Yeah, I think that we saw in our serum replacement experiments and also with BSA-ITS insulin experiments, we saw that BSA is a very strong carrier molecule, which can help because it's also a smaller molecule. It's also in the size of lipoproteins and high density lipoproteins range. Like for example, albumin is one of the good transport mechanism, which a lot of microRNAs bind to that. Like for example, when you see in the free floating microRNA, free circular microRNAs, they're normally in the biological fluids, you find free circulating microRNAs. But these kind of microRNAs are binded always to the lipoproteins or go to molecules, or also I observed specifically it binds to albumin or BSA molecule, so it can helps to the transport mechanism.
Okay, good. The other question that I had has to do with the arms of the precursor, so the 5' and the 3' arms. And I know that one of the papers that you showed that kind of led you down this trail was looking at 5p, whereas you ended up looking at 3p. And so I'm wondering if you did some analysis of the two arms and if you found that they had some conserved functions as far as their targets go. So in other words, have these things sort of, the two arms co-evolved? Well, obviously they've co-evolved, but have they evolved to have similar functions? And I just wanted to ask about what you might have done with that.
We did not focus on that level actually. So but that's an interesting question here. We did not focus on the 5' end, but we did in the transcriptomic analysis. It's all on 3' end analysis on that. But on the microRNA level, we never looked at on that. But that could be also interesting on the doing more on the analysis. Yeah.
sorry to bother you. It's more a curiosity.
Yes, Carolina.
Completely different the idea of culturing this type of cells, this type of organoids, let's say. So I was thinking at the human model, if you are culturing these organoids in grouped culture, and you will find that this microRNA will be a good predictor of the ability of the blastocyst to attach to the of embryo to attach. How you can, let's say, decide which embryos was the one that was producing a lot of this microRNA? I don't know if I'm
yeah, we can do yeah, we did also in this paper, like we checked microRNA qPCR experiments on the blastocysts, individual blastocysts, and all individually cultured blastocysts, and also in the group culture blastocysts. We saw a higher expression of this microRNA in the blastocyst itself, not in the conditioned media.
Okay, so so
you can
single
in
the
the
single blastocyst, you can.
Yeah, even in the single blastocyst, there is a high expression. So we could see a difference there. When we cultured the embryos individually, you can make a difference out of it. But yeah, that's what interesting that we saw, that these microRNA 378 is all highly expressed in the blastocyst and less expressed in the non-blastocyst. I'm talking just not the conditioned media, just the blastocyst. And also in the group culture, when they're grouped, and then we separated the blastocysts and degenerated ones, and we did the X-ray, and there is a high expression of 378 in the blastocysts. Yeah.
Okay, yeah, I was.
That's how that's how you really judge the biomarker there too.
Okay, good. Yeah. So we have a question. Mai, you can unmute yourself and ask the question directly.
Yeah, hi, thank you. That was a beautiful presentation. We work on microRNAs and vesicles as well, and this is a question that bugs me all the time, and people ask me, so I'm going to go ahead and ask you. And that is, how do you know of your microRNAs in your vesicles that are circulating or that are out there? How do you know that they're functional? How do you know if there is enough microRNA to perform the function that it needs to perform? And it's something that I struggle with as well. So
yeah, that's a very interesting question, and also like a intriguing question, I'd say. Yeah, definitely. That's what we also tried to figure out. Like we tried to show in this experiment where we did an EV treatment, whether this EV microRNA that is originated from this specific EV origin or not, by doing protease and RNase treatment. And we did also qPCR expression analysis. How, like, yeah, I would suggest like taking a minimum amount of EVs first and try to figure extract it from the conditioned media data. For example, I take the blastocyst EVs and then also non-blastocyst, and you do a microRNA qPCR expression analysis, and then you can determine how much expression levels are showing. Of course, determining functionality, whether this shows a functionality or not, it can be done only through the functionality experiments. Until you test them, you don't see it. But of course, when you are testing, you are doing higher concentration. You are taking from we ordered from the companies or you design your microRNAs, and we add or supplement to the cells. Then you directly test higher concentrations. But we really does not know. That's still, I think, need to figure it out. We does not know how much content of microRNA is available in these EVs per se. I think that answers your question, I think.
Thank you.
Thank you, Krishna, for the presentation. It was really, really amazing. Thank you everyone for participating to this EVIC lab. And I wish you a good day or night or whatever time is in your place. So thank you. Bye to everyone.
Thank you.
Bye. Thank you so much. Bye.